thank you all for having me. My name is Brock Burgess. I'm a research scientist at Fisheries and Oceans Canada, and I actually work just down the road at the Pacific Science Enterprise Center, which I'm sure a lot of you would be familiar with. So a lot of the work that I'm going to be sharing today is local and very relevant to a lot of the status of Pacific salmon right now in British Columbia. So my name is Brock, and I will be sharing a little bit about my work on environmental DNA applications to Pacific salmon management. Before I begin, I'm just going to talk a little bit about Pacific salmon, which, given the audience, I know is going to be a little bit of a refresher probably for many of you, but I really do want to set the groundwork for why the work that I do is important and why we should really care about salmon and new and exciting tools that we can develop to actually better monitor all of the populations that are in need of monitoring. So as many of you probably know, Pacific salmon are extremely important here in BC and in other parts of the world as well. But as scientists, we often break the importance down into a couple different components. We think about the ecology, the ecological reasons why salmon are important, from being an important prey species at all kinds of different life stages, whether they're eggs, whether they're juveniles, whether they're adults. They're important as part of food webs all throughout their lives. And they also have a tremendous value as within nutrient transfer between marine and freshwater ecosystems, given their life cycle and how they go out to the ocean and then come back to freshwater, but also just the aquatic to terrestrial environments as well. There's a large amount of nutrient transfer from the water into our forests, which are extremely helpful for a large number of our forest species, but just as the ecosystems at large. We also talk about the cultural reasons why salmon are incredibly important, because many of our indigenous people share very close spiritual relationships with these animals, and they also use them for sustenance as well. And then, of course, we have to talk about the economics when we talk about salmon, because they are extremely valuable, not only for the government and for our economy as a country and as a province, but also for recreational fishers as well, and fisheries within indigenous communities as well. So it's kind of the three major things that I usually have to talk about to people when I tell them why salmon are important. And so Pacific salmon have tremendous variation in their life history, which is really important in the context of the project that I'm going to share today, which is about the life history of salmon. And I don't want to dive too deep into this because I'm sure many of you are familiar, but salmon, they lay their eggs in fresh water, they hatch, they grow up a little bit, and then they go out to the ocean when they mature, and then they become adults out in the ocean, and they stay 1, 2, 3 years depending on the species, could be longer. And then they come back, they lay their eggs. Most species here, they then die and do it all again. And so I'm going to show how environmental DNA can be used to actually monitor salmon at specific different life stages. Unfortunately, Pacific salmon are in decline, or many populations are in decline. And this image here on the right is a result of the Pacific Salmon Foundation's 2024 publication, where they assessed populations of salmon here in the Pacific Northwest. And so the green fish, so the ones above that dotted line, those just represent populations that have shown an increase in total abundance since the last assessment, whereas whereas all the redfish show the opposite. These populations have actually shown lower numbers in total abundance since the last assessment. And so why are salmon in decline? We have so many initiatives out there. We have this festival. We have all kinds of funders putting money towards Pacific salmon, yet we still can't seem to stem some of the losses that we're seeing in certain populations. Well, there are a number of reasons for that. Climate change, pollution, barriers to accessible habitat. That's a really big one that a lot of people don't often think about, but there is a ton of habitat that's really high quality for salmon, but they just can't get there because maybe there was a dam put in or some sort of infrastructure that they no longer can reach some of the historical areas that they used to go and spawn. And of course, there's destruction of habitat and overfishing. So what's one thing that all of these reasons have in common? Well, they're all human driven, unfortunately. And so I think we do have a responsibility to put in all the effort that we can to try and help some more of these populations turn to the green side. One thing I do want to point out that's extremely important is all of these gray fish at the bottom. And so what that means is there just wasn't enough information out there for them to actually, for the Pacific Salmon Foundation to provide an assessment of their population status. And that's really important in the context of the work I'm going to share. So why do so many populations just lack the information that we need? Well, because salmon disperse everywhere. They have thousands and thousands of streams within the province that they can disperse into once they come back to spawn. And this presents a major challenge for management who are trying to find out how many fish are there from year to year. Some of the conventional methods of salmon enumeration, and I'm sure many of you are familiar, but things like stream walks, where you just go and you walk a stream and you look and you direct count. You say, okay, 1, 2, 3 fish as you go. There's other methods, fish fences, which are a little more accurate, but a lot more work involved to set up a fish fence where you can count them one at a time as they pass through some sort of barrier. Snorkel surveys, drones, electrofishing, trapping, there's all kinds of approaches to look at how many salmon there are at any given point. But they all involve a lot of work, and we just don't have enough people out there to send them to these thousands and thousands of streams and rivers to provide these up-to-date estimates of how their populations are doing. Enter environmental DNA, and that's where my work comes in. So disclaimer, I'm not a salmon biologist. When I started this work, I had never worked with salmon before, and so I've learned a tremendous amount about these fish since I've started. And so I'm a geneticist, so I got hired to lead these projects on the genetic side of things. And one of the ways I present myself as a scientist is that I want to build tools that can be used to actually impact whatever species that I'm working on. In this case, I'm working on coho and chinook primarily, but I want to actually do research that's going to matter. I don't want to just do research, publish it, and have it sit on the internet and nobody ever do anything with it. No, I want to develop a tool that managers can take, go, apply it, and actually have some sort of positive change. So how many people, before I continue, have heard of environmental DNA before? I see a few hands. That's good. I'm not surprised by that because environmental DNA is something that is relatively new in the sense of science, and by that it's about 15, 20-ish years since its conception as a field. But really, we've learned that all living things are constantly shedding eDNA, or shedding genetic material into the environment, which we call environmental DNA or eDNA. And so we're doing it right now. We have hairs that are coming out. We have skin cells that can be sloughed off us when we move. And so this is all entering our environment all the time. And it's no different for fish in an aquatic environment. And so some examples, again, of different types of, different sources of genetic material, things like tissue, your blood, our hair, feces, urine, sperm, eggs. And sperm and eggs are particularly important in the context of this project that I'm going to share, because when we talk about spawning fish, there's going to be a lot of eggs and there's going to be a lot of genetic material that are entering these systems during spawning. So technology has advanced to a point now where we actually have the ability to target specific fragments of DNA that's in the water, and we can actually use that to learn more about the organisms that eDNA came from in the first place. And some of the most common and conventional methods of eDNA right now are, simple species detection. So for example, I could go to the pond over there. I want to know if there's a certain species of duck that's been in that water. I could go take a water sample, take it back to the lab, and run my eDNA analysis, and then tell if that species was there or not based on the water and the DNA in that water. I don't even have to see if that duck was there, but I could tell you if it was there based on its DNA being in the water. And then, of course, there's biodiversity assessments, which is very similar, except instead of looking at one species at a time, you can look at thousands or dozens or a couple, it doesn't matter, but multi-species approach using similar technology and similar methodology, but it's just another really good way to do biodiversity assessments without actually having to see all of the different animals or organisms that you are actually looking for. And then finally, there's this idea of using eDNA for relative abundance, which is a major hot topic right now and will continue to be a hot topic because we're kind of, as a field, pushing that forward within environmental DNA research, where relative abundance is, the idea of being able to look at how much DNA is there and pairing that with a known number of your organism to try and find a way to use eDNA to tell how many fish are there or how many ducks were in that pond, rather than whether or not they are there, yes or no. Much more tricky, and not one that I'm going to dive a ton in today, a little bit, but just one of the other major interests within the field right now. And so a typical environmental DNA workflow is, within aquatic systems at least, you could have have a river or a stream or a creek or a pond, doesn't matter, or the ocean, looks like in this example. But you need to take some of that water, and so you can do that directly at the source, or you can filter that water directly at the source, or you can take it back to the lab and do it in the lab. But basically what you're going to do, you're going to take your water, you're going to filter it through a special piece of filter paper that's going to collect all that DNA. So all the DNA that's in your water is going to stick to your filter, and that filter then gets taken back to the lab where you can actually isolate all of the DNA that was on that little filter. And you basically condense your liter of water that you initially filtered on site all the way down into a tiny little tube that has pure DNA in it, and it will just be a few microliters, so a very small volume of liquid. So we can take that DNA in solution, and then we can, by using our highly sophisticated genetic tools in the lab, we can match up known sequences of DNA for our species of interest. We can match them up with what's in that little liquid, or that little tube that has a small amount of liquid in it. And so after we do that, we amplify the DNA using a process called PCR, which I'll talk a little bit about later. And then we can analyze the genetic fragments on the computer and match them up with known genomic databases of all these animals. So all these animals, or a lot of them, particularly with salmon, we have known genomes, which is the entirety of their genetic, it's basically their genetic signature, and we can match up what we find in the water to these databases to tell you what's there or not. And in this example, this was that biodiversity example where they're looking at what species are there, not just whether or not one is there or not. And that brings us to the project, the main research project I'm going to share with you guys today. And so our research question was, do patterns of environmental DNA, the amount of DNA that's there, does that reflect the temporal differences in salmon occupancy? And we used a couple different species to investigate this question, Chinook and Coho, because they have known differences with things like their out-migration timing and when the spawners come in. So they'll have slightly different windows of when they come in. And so do does the eDNA reflect that? Is eDNA pretty much the same all year round and just kind of building over time? Or are there big differences depending on, like during spawning, for example, when all the fish come in, do we see higher amounts of eDNA at that time? And these are questions we wanted to ask. And so our hypothesis, what was at the beginning of the project, was that eDNA concentrations would indeed be highest during times of spawning. And as that might seem fairly obvious because the fish are bigger fish, probably going to shed more eDNA, but also just given that specific life history event of spawning, they're actively releasing a lot of genetic material into the water at that time. So what did we do exactly? Well, we did a lot of field work. We had 2 field sites established. One was Slessy Creek, way out in Chilliwack. It's just by Chilliwack River Hatchery. And then we also had a site at Chehalis River, which is right by Chehalis River Hatchery. And you can see an image of those 2 systems on your right. And so, as part of the study design, I needed to visit these sites a lot. I wanted to capture the entire year of eDNA. And so what we ended up deciding to do was that I would visit these sites 3 times a month, every month, and these days would be in a row, so a Monday, Tuesday, Wednesday, and I would do that every month for a full year. So 36 visits to Chilliwack, 36 visits to Chehalis River and Agassiz. And each day, what I did was I collected 3 different 1-liter water samples from these sites. So I went to Chehalis River first, then I went to Slessy Creek out in Chilliwack. And so each day I had 6 samples in total between the 2 sites, and I did that every day, 3 days, 3 days, 3 days a month for 12 months. 216 total water samples that I had for eDNA analysis. And so what does that actually look like? Well, on the lab side of things, there's that process I mentioned called a DNA extraction, which is really important. It's where I take all the DNA that was in the water that's now on that filter paper. I just want to get the DNA from the filter paper, concentrate it into a small volume of liquid. And then I can actually build what we call assays, which are basically targeted a targeted method of amplifying whatever DNA we want, essentially. And we do that. I can design that before I even start the project. I know I want a little fragment of DNA that's going to amplify coho, and then I can get a separate one that I know will amplify Chinook only. So they won't amplify any DNA from each other. It'll just be coho only versus only Chinook, and that way I can compare between the 2 species how much DNA is being released into the water from Chinook versus from coho. And this is all done using a process called qPCR, which you may or may not have heard of, but that's quantitative polymerase chain reaction. I don't think I'm going to bore everybody with what that means and the technical level, but it's simply just a method of amplifying DNA in a way that allows us to see how much was there in the first place. So we can use that technique, and I see, oh, there's this much coho DNA, this much Chinook DNA. That's all you need to know. So just to refresh everyone about what the research question was, we wanted to see if the patterns of how much eDNA is there actually reflect the differences in life history in terms of which fish we expect to be there and how much of them do we think should be there. All right, so to answer that question, yes, environmental DNA absolutely did reflect these temporal differences in freshwater habitat between Chinook and coho. And on the right, you can see coho. It's very nice, it's very neat and tidy looking graph where we can see, and I'll add these red blocks to the bottom. Those are the kind of expected peak spawning windows for these 2 systems, and these were provided by the local hatcheries there. And so you can see that, yeah, absolutely for coho, for example, on the right, the highest concentrations of eDNA were definitely during spawning. It was the same for Chinook, but it's a little bit messier, and we can talk about why that is. But before I go on, I just want to point out that this y-axis here is not linear. It's goes up by tenfold every little increment. So 1, 10, 100,000, 10,000. So just because there are slight little differences what it looks like in terms of where the boxes are, there can be actually a big difference in what that concentration actually is. Because, for example, this point up here, 10,000 is extremely high, and these ones down here are only closer to 500 or 100 or so. So, it'd be much big differences. Okay, so why is Chinook a little bit messier than Coho? Well, there are a couple reasons for that, and so it might be because Chinook don't have as clear of a coho. They have more variation in how long they can spend before they outmigrate. How about that? In coho, that's generally known in these two systems that we studied. That they leave all at the same time. After they reach a certain age, when they're juveniles, they leave to go out in the ocean, and they have a very tight window in which spawners come out in October, November, December, when spawners come back from the ocean. Whereas Chinook actually have a couple different Chinook migrations at these systems in particularly in Slessie, where there's an early Chinook run, and there's not clear distinction between when that run stops, when the other one starts in the fall. So the trend doesn't look as clear over there for Chinook, which is expected. And then the other, another really important thing to point out is that the only months that we didn't detect any DNA from one of these two species at all is right here in the winter, where we didn't actually find any Chinook eDNA in the winter at Chehalis. And that can be for a number of reasons, but it could represent, based on conversations with the local hatchery managers, that there was only eggs there at that time. There was a period where there aren't any fish that have emerged yet, whereas coho might stay around a little bit longer and always be present at some age in these systems, which is why we didn't ever have an absence in the coho relationship. So it did match expectations based on conversations with the local experts about what these fish are, when these fish are actually there. And the last thing I want to point out about these figures is that there's a major outlier, which was really interesting when we saw it. So I want to point you guys out to May. May specifically with Chehalis, and you can see that this is quite a bit higher than all of the adjacent data points, and it initially really threw me off. I was like, "Wait, what? Did something go wrong here? Why are we seeing this large spike in eDNA in May?" And so I dove a little deeper into that. You might recall that I said I sampled 3 days in a row each month, and so this is the same data that we were just looking at, except instead of averaging the amount of DNA and putting them into just by month, this time I show you the breakdown day to day within each month of how much DNA was there. And it's important because we can see that it was really just one day. One day in May was responsible for this really high outlier here in the monthly relationships. So what is going on there? Well, it required a conversation with the hatchery manager, because I had to show him this and be like, okay, well, what is going on here? This doesn't make any sense. And he told me that morning, so the morning of the day that I sampled, before I got there, they had done a release upstream, 75 meters upstream, 150,000 smolts were released. And so that was really validating, though, because, and it actually turned out to be a really important component of this work, because what that tells us is that all of the eDNA from 150,000 fish was almost essentially flushed away after a day. Those fish left immediately, which is what was expected because they were smolting at that point, and we didn't detect it over the consecutive days, which is really important when we think about eDNA as a tool for fisheries management, because, well, we hope that eDNA presents a snapshot of what's there at the time in which we sample. If the DNA just builds up continuously over time, then it really would make it not as useful as a tool for helping management, if that makes sense. So this was really important to show that even if there is a ton of fish at one time, well, we're confident that when we see our eDNA concentrations, it's going to be representative of what was there, the few hours before that we actually sampled, which is really important. So overall, of course, we know that salmon are really, really important. And we talked about those, ecological, economic, and cultural reasons. But unfortunately, there are populations, many populations, that still are in decline. And we need more efficient population monitoring techniques, because that can help us to actually get to all of these different, well, maybe not all, maybe not ever all of them, but we can certainly reach more of the major systems if we have these quick, non-invasive tools like eDNA, where you just have to go in, collect some water, and leave. That can be a lot quicker than doing a stream walk or setting up a weir or any other method, whether it's drone surveys or snorkeling. So eDNA is really advantageous for fisheries management for that reason specifically. It's a quick in and out, non-invasive. You don't have to see the fish, don't have to bug the fish, don't have to touch the fish, stress them out. It's a non-invasive method. It's quick, easily scalable to lots of systems. And that's what I showed here with this project, where we showed eDNA, demonstrated it as a versatile tool for fish monitoring, and a lot of these techniques can be extrapolated well beyond Pacific salmon as well. And there's all kinds of other applications in which eDNA is being used for and being explored for right now. I wish I had more time to talk about a lot of my other projects, but one in particular that I am working on and have built is a eDNA tool to look at relative abundance across streams. So for example, I have another project where we went out to 40 different creeks, all mostly within Metro Van, and I sampled at 40 different creeks, and I built a model that accounts for environmental variation at each site, because there will be things in the environment that change how much eDNA is there and whether or not it gets moved. If you think, okay, well, DNA is still a physical thing, so if we think about discharge or water flow, a higher flowing system with more water is going to move that DNA more quickly away from whatever the source. And so by accounting for things like discharge, temperature, turbidity, I was able to actually build a model that showed it's very promising that we might be able to use eDNA as a method to provide a relative estimation of the number of fish that are actually there across different sites. So lots of excitement still within eDNA, but like I said at the beginning, it is a relatively new field. 15 years doesn't sound new, but in the terms of science, that is very new. And so I think that eDNA is going to be a major component of fisheries management. I mean, it already is becoming a major component of fisheries management, but that's only going to continue as time goes on. Projects like these require a ton of work. This was almost a 2-year project within the planning. Well, yeah, by the time the planning and the paper actually got written, it was over a 2-year project. And there were dozens and dozens of people that came out in the field with me to help with all of this data collection over that time. People from Pacific Science Enterprise Center, Cultus Lake Labs, Oceanwise, the Chehalis First Nation themselves, and then the 2 hatcheries that I mentioned. But this project was funded, and this is really important, it was funded by the Pacific Salmon Commission, as well as the Pacific Salmon Strategy Initiative within DFO. So there's a ton of people that I have to thank for this work, and including you guys for sitting here and listening to me today. So if anyone has any questions, I'd be happy to take them. So her question was that she recently saw that we're in the midst of, or at least in August, was one of the largest salmon runs in recent history. Well, I don't exactly know how that ties in with these specific sites that I went to, but what I can say is that when we read in the news that there's a big salmon run, for example, well, my question becomes, well, where and what and when? Because there's all kinds of different species, and their population sizes and their run sizes will differ completely based on where they are, whether they're up in the Yukon or whether they're down in California. And this was specific to Vancouver. Yeah, so I don't know how that would have matched up with, projects like mine. But like I said, I'm not a salmon expert, so I can't answer your question better than that. There absolutely would be a lot of salmon DNA in the ocean, but where that DNA is at one point, I mean, that's a great question. So, for example, if you just went down and took some water at the beach there, would there be salmon DNA? Well, I'm not sure. There might be, but there might not be, because that eDNA, once it's in the environment, it starts to degrade. So the UV light, the environmental, like there's microbes, microorganisms in the water that literally eat DNA. That's something that they consume, and so DNA is being broken down as soon as it's off our bodies, and so that's why. eDNA is a useful tool as being a spatially explicit tool. It shows us what's there specifically. So if there's not salmon around, the beach where you sample, like I wouldn't necessarily expect there to be DNA, especially given how diluted it would be in the context of the ocean, if that makes sense. That's another great question, and one that is currently being explored in the literature. And so she asked about, where in the water column do we sample, and does that matter? Well, generally, when you're designing an eDNA study, you want to sample as close to where you think your organism is going to be. And so, if we expected the fish to be more at the bottom, then okay, maybe a better alternative would be to sample straight from the bottom. But and so for our project, we sample at the surface, always at the surface, just probably 5 centimeters or so under the surface of the water. And there was a reason for that. We want this to be a tool that is easy to use. And so if it requires a ton of different equipment and effort to go and sample water from, say, the middle of the river at the bottom of the river, for example, well, that's not really useful for management, because that's now, it's no longer being a really easy, accessible, versatile tool. And so part of this project was to sample right at the surface, which is conventional for this type of work. But we wanted to provide the easiest way possible to show that you can use this as a tool, because then it will be much more helpful. Absolutely, yeah. So every fish will have a different genetic signature. However, one of the beauties of using this technology is that I can look for a region of the genome of coho, so a region of their genome where all coho have the same sequence of DNA. So there's not different between them. I can target that region and then amplify it, knowing that there's differences within we call it the amplicon, but the region that I'm looking at that is different between the two, if that makes sense. And so I can use a wider area that's more conserved across salmon, and amplify that to look at differences at an individual level, and be able to point out, like, oh yeah, there's 6 different fish here, and these are who they are because they have different DNA. And so that's something that we absolutely can do, and is being explored in an environmental DNA context right now.
So you can say there's 10 coho here, and each one of them is different.
That's certainly a goal for the future. I would say right now people are really trying to make that happen on the lab side of things. I do think the technology is here. It's more about, can we figure out how to do it? And so people aren't doing that just yet, but it absolutely is something that will be the future. Yes.
This is where you're sharing information throughout the world. There must be other places doing some of this work as well. And
yeah, there absolutely are. Particularly, there's a lot of people in China and Japan who are doing this type of work. And Japan specifically gets certainly has their own Pacific salmon populations there. And so U.S., Canada, and then I would say some of the major East Asian countries have a large investment in eDNA research, and are absolutely be pushing it forward globally. Although I will say that the work that not this specific project that I shared, but the ones that I didn't share today are very much cutting edge, and nobody in the world has been doing some of the work that again, I didn't share today, but certainly would be happy to talk about it after with anybody who's interested in. Well, thank you all.